Professor Albert Fornace Jr. is director of the Water Center of Innovation for Metabolomics, Lombardi Comprehensive Cancer Center in Georgetown University. He's going to talk about integration of metagenomics and metabolomics in gut microbiome research.
Okay, well, thank you very much. It's great being back here again and seeing old friends. And I'm sort of going to talk about something different today than I've done in the past during my visits. And we'll be talking about some of our work where we're trying to merge or at least integrate some of the metagenetics with metabolomics in the gut biome. And this is a joint effort. Miriam Gudarzi and my group has really spearheaded much of this work, and also Heng-Hong Lee has played an important role. And it's also done in collaboration with John Braun's group at UCLA, who are famous for some of their microbiome work going back a ways. Okay, so what I'd like to do today is just briefly give an overview of microbiome and health and disease, talk about how we can now assess these bacterial communities, many of which you can't grow in culture, using high throughput sequencing, and then try to tie this in with inflammatory bowel disease. It's a big problem and increasing in frequency. And then I'll make some introductory comments, very brief, on metabolomics and what we've built at Georgetown and developing microbiome metabolism approaches. And then we'll go down the impact of IBD, namely inflammatory bowel disease, on metabolism. And then I'll finish up with a more acute model for injury to the gut and the microbiome using ionizing radiation. There is some overlap with other types of injury agents, but I'll just show this as an example. So in the case of the gut flora, it's highly complex. And let's see if I can find the mouse. Probably not. It's highly complex, and as I've indicated here, there's 10 times more bacterial cells than there are both germ and somatic cells in humans. Also, the microbiota are plastic, and there's a variation between individuals, and it can change with time. As far as advances in understanding the microbiome, next-gen sequencing and the like has shown that there's probably about 10,000 different microbial species there. There's 150 times more genes if you add up all these different genes from bacteria than there is in the human genome. And one approach that we've been using is using 16S ribosomal DNA sequencing for rapid assessment. And a term you'll see in the literature, and also I'll be using, is OTU, which is for Operational Taxonomic Unit. And so there's a tremendous amount of effort in this area. If we look at human gut biome publications in PubMed, you can see that they've really shot up over the last recent years. And as far as trying to tie the gut and microbiome interactions, I'm showing an example here of symbiosis of potentially beneficial or commensal bacteria. And what I'm showing here are receptors or surface proteins that are glycoproteins, so they have sugars on the ends. The red triangles, which I can't point to, would represent fucose. And so there's a very common null mutation in humans where the enzyme that hooks up the fucose to these proteins is lost. And so what we would have here is, if we think of the red triangles as the fucose, there's a variety of bacteria binding here, and some of them will have commensal or potential beneficial effects. There's also some advantages, particularly, There's also some advantage in not having the fucose, for example, with certain pathogens, but we won't be talking about that today. And an important point is that these FUT2 null, which we refer to as non-secretors, are at a higher risk of inflammatory bowel disease. So as far as OTU, just very briefly, it's based on 16S RNA sequencing. It's present in all the bacteria in the archaea. And so what we basically do is that we're using the conserved regions to prime, and then we're sequencing a short stretch of about 150 or so nucleotides. And then we define these taxa of related bacteria by more than 97% identity. And so you get these groupings of bacteria that are somewhat related. And so you can start looking at the different families or genuses of bacteria that, and how they change in disease or injury. Okay, is getting one to inflammatory bowel disease, this symbol up here on the top right shows that there's multiple components that are contributing to inflammatory bowel disease. The incidence is increasing almost worldwide, both in the USA and Europe, Mid East, China, and other regions. Obviously, the genetics have not changed that much in these populations. So there's environmental effects. Many feel that it's dietary effects as well. In the case of inflammatory bowel disease in Mideastern children, there appears to be, from a variety of studies, a higher prevalence, particularly recently. I should also mention, before getting into just the Mideastern story, that we see increasing incidence in a variety of different countries and ethnic groups. In the case of children of Mideastern descent, we see a more pronounced or higher disease activity level for Crohn's disease, indicated by the gray. And what I'd like to do is focus really on some of the genetic components that influence inflammatory bowel disease and not go into the diet and environmental issues today. And I think one interesting study that came out of Australia was they looked at the incidence of inflammatory bowel disease in a pediatric population, and so obviously the diet and the environment is the same, and they saw a much higher frequency in this particular study. So it again highlights that there's genetic components as well as possibly gene-environment interactions. Now, going on to metabolomics, where we're going to try to link the microbiome and the genetic approaches in microbiome research with metabolomics, is these are well-developed omic technologies or platforms. And in the last 10 years or so, there's been really an explosion in our understanding and ability to assess metabolites at the omic level. And so the idea here is that we can now develop a integromics type of approach, which has very challenging bioinformatics, which I won't have time to speak about today, to develop sort of a more holistic systems medicine approach using these different omic platforms. And just to give a plug for the metabolomics, it really provides a snapshot of current physiology. Subtle changes in RNA or protein levels or activities can oftentimes give you large changes in metabolite levels. Just briefly, one of the reasons that metabolomics has taken off is that in addition to the earlier technology using NMR, which was very elegant and developed by Jeremy Nichols and others, the availability of these time-of-flight mass specs, or so-called QTOF mass specs, which came on line at least in academic centers about 10 years ago, has really driven the metabolomics field. And if we look at PubMed publications over here on the right, we can see that it's really skyrocketed in the last 5 to 10 years. Now at Georgetown, we really got in on the ground floor thanks to some of my interactions with Frank Gonzalez, the head of metabolism at NCI. And we had one of the first QTOFs at academic centers about 2007. And now we have a water center of innovation. This is one of the major manufacturers of these LC-MS, liquid chromatography mass spec type machines. We have over 40 faculty users, 20 outside users, and we're the metabolomics core for the Mid-Atlantic Cancer Center Consortium, including Virginia and Hopkins, Maryland, and other hospitals. And it's also been an important component to generate grant support for many of our investigators, both at Georgetown and nearby. So what we'd like to do then is develop a microbiome metabolic biomarkers for IBD. And so as a sort of a proof of principle, our first effort was really to say, can we see differences in the bacterial niches in the cecum versus the sigmoid regions of the colon? And in collaboration with John Braun's group, we had lavages from patients undergoing a routine colonoscopy. And what we can see here over on the right is that if we look at these OTUs, there's differences in the bacterial spectrum for cecum versus colon. Over on the left, we're looking at a heat map with the darker colors indicating higher levels of particular metabolites. And we can see a subset of metabolites much higher in the sigmoid, and then others that are higher in the cecum. So we are starting to try to piece together then the connections between the metabolites we're seeing and the bacterial makeup of the gut in various regions of the gut. And you can play these types of games and develop a variety of correlation and type of associations. And here we're looking at individual metabolites and also different OTUs or bacterial groups that are coming up and looking at the connections. And I think the take-home message is that between the cecum and the sigmoid, we're seeing a different pattern. And what we've done is pooled all of our human samples and then tried to do correlation analyses in these pooled samples. And when we looked at proteomics done at UCLA versus the genotyping done primarily at UCLA, we see some interactions becoming apparent. However, if we look at the bacteria versus the metabolites, we see this far, far richer interactome of bacteria and metabolites that are coming out. And so there is really a striking correlation then between what we're seeing at metabolite level and the distribution of bacteria. So how does this all interact or affect inflammatory bowel disease? Well, as I mentioned to you, the FOOT2, we have the secretors and non-secretors. It's very common. Up to 60% of many populations of humans will have at least one null allele for the FOOT2, which is referred to as SE, lowercase. And if we look at the just doing a sequencing of the number of genes, which on the top right, which will give you an estimate of the diversity of, or the richness of the bacterial populations, is what we're seeing is that there's a richer population, more genes, more bacterial species in the secretors, namely the ones that have the FOOT2 and are able to have this fucose glycan on the cell surface proteins. If we look at the different types of pathways that are enriched different pathways involved in metabolism, we're seeing differences in the secretors versus particularly the null. And then these are particular metabolites showing the increases, particularly for the non-secretors. And the interesting thing for the bottom lower half of the slide is that we saw essentially the same profiles in FUT2 knockout mice. So the FUT2 knockout mice develop inflammatory bowel disease, and the patients with the FUT2 null phenotype are at increased risk for inflammatory bowel disease. So what about inflammatory bowel disease itself? We're just getting into this now. This is a pediatric population here, and we can see a clear separation by PCA analysis, looking at just metabolites from colonic lavages. We find a variety of metabolites coming up. I don't have time to really go into individual ones. I'll show one or two examples further down. But if we look at a heat map of metabolites that show higher levels in healthy versus Crohn's disease in this pediatric population, we can see a clear separation. Now, one or 2 points about this slide that I wanted to make is that the healthy individuals here are family members. So we're trying to control for environmental differences and looking at unaffected siblings in most cases. This supports our hypothesis and going forward for more extensive studies that we're planning, is that there's a genetic network influencing the microbial metabolite composition, and the metabolites may in fact be creating a susceptible or pre-disease type of state for Crohn's disease, which is a common inflammatory bowel disease. This is a slide from John Braun, which just sort of summarizes approach using SNPs and the like to identify, for example, the FUT2, the FUT2 secretors and non-secretors, changes in metabolite levels, changes in microbial abundances, and then predisposition to chronic disease. Over here we have the genomic and proteomic levels from the host, the microbiomic level of differing bacteria, and then the metabolite level, which is really a mixture of the two. So in the final few minutes, I wanted to sort of go into a more simple, direct assessment of what happens to the gut microbiome with injury. And here, what we're trying to do is develop a integrated approach to assess a particular injury agent, ionizing radiation, which is known to produce marked toxicity in the gut since they're rapidly proliferating cells. And so the idea here is to say what kind of metabolite changes do we see, and what type of changes do we see in the microbiome. And the take-home message is that we see some clear separations even at non-toxic doses of radiation, so that we can see differences in the metabolite spectrum with this PCA plot control versus a few days or many days after radiation. And we see, and this is a whisker plot here, with either a non-lethal dose or a lethal dose of radiation at 3 days, we see a marked reduction of this particular pneumococcus bacteria species after radiation. So we're seeing both changes in the bacterial composition as well as marked changes in the metabolome. And Hang-Hong Lee and my group carried out many of these studies, and this just sort of shows the approach we're using. We're not doing endoscopy, we're just doing metabolomics and sequencing of the feces. But what we can see is just looking at these OTUs, or groups of bacteria, is what we can see is a complex distribution of these sort of general groups, and 3 days after radiation, even with a non-lethal dose, we see a marked change in the composition of the microbiome based on the 16S sequencing that we've carried out. Okay, in the case of metabolomics, all these red dots here indicate either metabolites that are decreasing in abundance in a significant way or increasing in abundance in a significant way. And I've already shown you a heat map, but this is another one showing a separation between control and irradiated mice. And again, at the non-lethal dose, we can see several days after radiation, there's a rich heat map showing differences for both bacterial metabolites as well as host metabolites. In the control, these are reducing, red high, green low. After radiation here, there are lower levels and going up after radiation. So this gives you, from a practical standpoint, biomarkers for radiation exposure, which could be important, for example, in some type of radiation accident or episode. If we do some of the pathway analyses based on the types of metabolites that we see coming up and metabolite pathways, I've listed some here. All of these are separate stories, but I'll mention that we see a significant perturbation in bile acids secretion. And this is highlighted by individual metabolites. I've shown some metabolites here over a time course, but let's just focus at several days after either the low or high dose of radiation. We see these primary bile acids increasing in the feces after radiation, whereas this secondary conjugate, or a deconjugate actually, bile acid actually decreases after radiation. And so this is due to the fact that at least our working model is this is due to the fact that some of the bacteria that are known to do this deconjugation are reducing in levels after radiation. If you try to develop interaction maps looking at the changes in the bacterial spectrum after radiation, and then looking at changes in metabolite levels after radiation. So we have the microbial taxon in blue and the metabolites in yellow, and then we either have negative or positive correlations. You can see a rich sort of interactome map that's coming out. For example, we're seeing some, in the case of some of the, and I don't have time to go into this, that some of the bacteria that are involved in bile acid deconjugation are actually going down along with these metabolites, such as I showed on the previous slide. So then just to summarize then, is what we've tried to do here primarily is to develop a workflow to assess the impact of the microbiome or metabolism in the microbiome, and develop biomarkers to assess both the course and the severity of disease or injury. In fact, in the case of inflammatory bowel disease, we have perturbations in both bacterial makeup and metabolism. With inflammatory bowel disease, we have obvious interplay between genetic and environmental factors. And then in the case of the FOOT2 effects, we can show that there's changes in the gut microbiome as well as also in the metabolism of the microbiome by losing this particular host glycan modification. In the case of responses to bacterial injury or radiation injury, we see profound effects on bacterial populations, and some of these so-called beneficial, or at least let's refer to them as commensal OTUs, such as the rheumococci and several of the others, we see them going down after radiation very rapidly. And so changes in some of these metabolites, such as the bile acids, may reflect a reduced intestinal absorption, as well as lower rate of deconjugation, for example, by the reduced levels we see in rheumococcus. So I think that what we've done at this early stage is set the stage for having the tools available to start looking more extensively at inflammatory bowel disease and other injury responses that are going to impact the gut, and assess the myriad of changes that are occurring in these 10,000 odd species that reside in the gut microbiome. So I'll stop there and thank you for your attention.
Thank you so much for the presentation. We are running out of time, but I think there is still time for one question or two questions. Is there any question?
Dear Professor, thank you for your excellent presentation about hereditary bowel disease. My question is, when we are going to do case control studies or genomic association studies, we are comparing genetic polymorphism between these individuals and controls. Sometimes we have a certain genotype is sharing between the diseased one and the controls, and the questions like, for example, the pathogenicity of type 2 diabetes and the genetic factors contributing to it. Is this sharing between the controls and these individuals, for example, like type 2 diabetes as multifactorial disease, but the control have not the disease in spite of they have the same genotype? So my question is, metabolomics does have a role in the pathogenesis of multifactorial disease like myocardial infarction and type 2 diabetes or not?
Yes, it's sort of difficult to hear with the PA system, but the metabolomics is essentially applicable to any condition that affects metabolism, which means that we can essentially use it for almost anything. I think in the case of the gut, we've published a bit, and people at Georgetown have published more, developing using metabolomic approaches to assess for responses to therapy, for example, GI cancer and the like. Also, metabolomics is being used for GI as well as other types of tumors as early biomarkers. I think that for any disorder, whether it's infectious or neoplastic or autoimmune, that's impacting the gut microbiome, we're going to see rich changes in the metabolome, and I think that it's going to be applicable to additional areas. It's probably with the fact that we're running a little late, it's probably best to discuss this afterwards without the PA system.
Thanks so much, Prof. Fornace. The next speaker